Biomolecules are compounds that are important for life to function. Bio meaning life, and molecules are molecules. So there are many different types of biomolecules, however the following are most important as described by the Lumen study plan. There are proteins, nucleic acids, lipids, and carbohydrates. For time purposes, we will only be focusing on nucleic acids for this video. So what exactly are nucleic acids? In layman's terms, they are the thing that carries the blueprint of the cell and carry instructions for life. So because this is a review, can anyone tell me what they think that nucleic acid is? What type of nucleic acid is in that picture? I'll give you about 3 seconds. All right. If you had said deoxyribose nucleic acid, then you are correct. So there are 2 types of nucleic acids, deoxyribose nucleic acid and ribonucleic acid. And these are 2 very different nucleic acids, and they both serve different functions. So deoxyribose nucleic acids, otherwise known as DNA, can be structurally determined by the double helix feature that it has. A double helix being the classic DNA shape that we see in this picture here, where it seems like there's 2 strings and they're twining around each other. Then, of course, there's ribonucleic acids, in which they are much smaller than DNA has a tendency to be, and it has a singular helix structure. Now, of course, DNA and RNA, nucleic acids in general, are made up of nucleotides. So, nucleotides are made of a nitrogenous base, a pento or 5-carbon sugar, and a phosphate group. So, the nucleotides for DNA are adenine, guanine, cytosine, and thymine. RNA has adenine, guanine, cytosine, and uracil. It is very important that one does not get these 2 mixed up, as it can cause some trouble or problems when you are trying to transcribe DNA into RNA and vice versa. So a little bit more information on RNA. RNA has many different types of functions, and because of that, they all have different names. So messenger RNA is transcribed as mRNA, tRNA is transfer RNA, and rRNA is ribosomal RNA. And that is the end of our slideshow. Excuse me. Thank you for listening, and I am excited to see what else we will learn so far in the semester. Thank you. Bye.
Hi everyone, my name is Eric. I'm here to talk to you guys about atomic bonds. They're an essential part of biology that I think is important to learn about, so hopefully you guys can learn a little something from my little presentation. So what exactly are atomic bonds? Well, they're just bonds between atoms. They come and bond together to form everything that we see in life. Trillions of atoms bonding together to form everything that we see and touch and feel and smell and all that good stuff. So why are they important? Well, like I just said, they come together to form everything that we see and need in nature. So such as water, just 2 hydrogen and 1 oxygen bond. Multiple molecules of H2O bond together to form what we see in water and what we drink and what we need in life. So it's actually different kinds of bonds that I wanted to get into. And the first one I wanted to talk about was ionic bonds. And in ionic bonds, it's important to remember the I. That's how I get to remember ionic bonds. Because in one atom is taking an electron and the other one is giving. And ionic bonds, one atom is saying I, ionic bond, need that electron. So it takes one and the other one is giving that to the electron. And what you get is ions, and they become positively or negatively charged from electrons because electrons are negatively charged particles within atoms. And with ions, you actually have 2 different types of ions if you give or take the electron. In cations, you have an atom that has given an electron, it's lost that negatively charged particle, making it positive. And the way I try to remember that is cation. Cats have pause sounds like positive, and cations are positive. It's a little silly, but it really helps me in trying to remember that. Now, on the opposite side of that, you have an anion, which is a negatively charged atom. And that one's actually taken the electron and gained it, making them negative. And the way I try to remember that one is anion. An is like anti or opposite, opposite from cation, making it a negative ion. Now, the other type of bond that I wanted to get into was covalent bonds. And this is when two atoms form a bond sharing electrons. And the way that I remember that is the co. And covalent is cooperative, or think of like co-manager, co meaning together. These are the ones that are found most commonly in living organisms. And there's actually two types of covalent bonds, just like there was two types of ionic bonds, and there's polar and nonpolar covalent bonds. And in polar, there's an unequal sharing of these electrons, and meaning that each atom will be more positively or a little more negatively, only slightly charged. The way I try to remember that one is polar. Something is polarizing, meaning it's in a disagreement or it's opposite of each other. So those are they're in a disagreement in the sharing of the electrons. But in nonpolar, they're actually shared equally between the atoms. So there's not that disagreement in sharing the electrons. Now, hydrogen bond is actually a subcategory of a covalent bond. It's a weaker bond. It's a weaker form of polar covalent bond when a hydrogen atom is present in the bond. And the best example I can give that is water. So in water molecules, which we know is H2O, two hydrogen atoms, and one oxygen atom. And in this bond, two hydrogen atoms actually become slightly positively charged, and the oxygen becomes slightly negatively charged. So that slight positive charge in hydrogen attracts other water molecules through the negatively charged oxygen, and it brings all the water together, and that's how you get the form of liquid and water. Water is the way they're formed together. So that was kind of the last bond I wanted to talk about. Hopefully you guys learned a little something from this little presentation, and thank you.
So today we're going to be talking about ion channel linked G-protein enzyme linked receptors, which are three different types of cell surface receptors. The first one we have here is the ion channel linked receptor. This one binds a ligand and opens a channel through the membrane, which allows specific ions to pass through. So right here, this gray area is the plasma membrane. This area right here, the blue part, is the ions, and this orange area is a channel, or in other words, I would like to say like a gate. So this gate would not open unless the signaling molecule or ligand is the same thing, goes to it and gives the signal to open the gate. So if the channel doesn't open, that means the ions are not able to pass through. The only way they pass through is with the signaling molecule, the ligand. So these ions move from a high concentration to a low concentration. So in the exterior is higher concentration. So their goal is to go through the plasma membrane so that they can be in a lower concentration, because in the interior is a lower concentration. So an easy way to remember this is saying that if you go to someone's house and they have a gate, the only way for you to go in is pressing the button and saying your name and stuff like that. And then they'll open the gate so that you can go inside their house. Otherwise, you wouldn't be allowed to go inside their house. And it's the same thing with this ion channel. The ions will not be able to go inside or through the plasma membrane if it's not with the signaling molecule or the ligand. So examples of this is sodium, calcium, magnesium, hydrogen. All of these go through this. As you can tell in here, right here, the extracellular fluid is, like we said, in a higher concentration. So the ion's goal is to go through the plasma membrane so that they can be in a lower concentration. Next one we have is the G-protein receptors. This one binds a ligand and activates a membrane called a G-protein. So there's a difference between the G-protein receptor and the actual G-protein itself. So right here I have the inactive area and the active area. So when it binds to a receptor, it's going to activate the G-protein. So an activated G-protein interacts with either an ion channel or enzyme in the membrane. So the binding is going to cause a change of the protein receptor, and because of that structural change, it will activate the G-protein, and the subunits are going to dissociate. So as you can see here, this is the inactive. So when it's activated, it's going to change the GDP to GTP. An easy way for me to remember this is like the ATP has way more energy than ADP. And this is how I remember. When it's active, it's GTP. I don't know if it's a helpful tip, but that's how I remember. So as you can tell here, they're together, the 3 subunits, but when they're active, they're going to dissociate. The beta and the gamma are going to stay together, and the alpha is going to stay by itself. So all this is like a cycle. They're going to do this all the time and just keep on going. An easy way to remember this is like saying a kid is growing up, eventually leaves home. The mom and dad are going to stay together. Right here, as you can tell, they're together. But then here's the kid, he's leaving the house, and the mom and the dad are together still. And that cycle is just going to keep on going and going and going. That's how I remember G protein. Example of this is botulism. This is a bacteria that is pathogenic to humans and can release poisons that interrupt specific G protein linked receptors. So this specific bacteria, it makes people lose their eyesight, they feel fatigued, they can't speak correctly, and that's because this bacteria releases poisons that interrupt G proteins. So yeah. Lastly, we have the enzyme linked receptor. This one is a cell surface receptor with intracellular domains that are associated with an enzyme. Right here is an inactive start place. Right here, as you can tell, this is the kinase. Kinase is actually an enzyme that transfers phosphate groups from ATP to another proteins. So right here, as you can tell, right here is phosphorylated, which means active. It's activated, in other words, as you can tell. So the activation of the enzyme sets off a chain of events within the cell that eventually leads to a response. And the binding initiates signaling pathways as well. So I have an example here with the tyrosine kinase. As you can tell, the ATP, like we said before, ATP has way more energy than ADP. So the ATP looks like it's going to the ADP. And right here is the tyrosine is phosphorylated, which means it is activated. And these are all the tyrosones and whatnot. So those are the 3 types of cell surface receptors. I hope you guys learned and liked it.
Transcription and translation. So transcription is the first part of protein synthesis. Protein synthesis produces amino acids, which are the building blocks for proteins. So transcription is the first part. Transcription occurs in the nucleus, and it can occur in the same time in prokaryotic. Transcription and translation can occur in the same time in prokaryotic organisms, but not in eukaryotic organisms. So what transcription does is DNA is copied to be in mRNA form, and DNA and mRNA are different. mRNA has the ribose sugar, and it's single-stranded. DNA is double-stranded. And RNA tends to be shorter. But RNA is able to travel outside of the nucleus to the ribosomes to engage in translation, the second part of protein synthesis. But in order for transcription to occur, DNA needs to be split, and helicase does this. And the transcription process, RNA polymerase is essential to the process. It adds the nucleotides, it transcribes them. And the thing about RNA polymerase is it adds the nucleotides from the 3' end to the 5' end. It can't do it from the 5' to the 3' end. So if you see DNA and it's from 5' to 3', you have to read it backward, because RNA polymerase can only transcribe from the 3' end to the 5' end. So you got to read it from 3' to the 5' end. And there are 3 types of RNA polymerase, and the one we have to remember is RNA pol 2. So there are 3 steps in transcription: initiation, elongation, and termination. Termination occurs when RNA pol reaches a termination codon. And promoters are needed so that our RNA pol knows where to start, where to begin. Yeah. So the products of transcription, sorry, that's spelled wrong. Transcription produces pre-mRNA, which is in the nucleus, and then it has to undergo some processes. And then it can move outside of the nucleus to the ribosomes, and this includes the 5' cap, 3 polytel, and the splicing of introns. Remember, introns are the ones that are not expressed. Exons are expressed. Yeah. And then translation is the second part of protein synthesis that occurs outside of the nucleus on the ribosomes, and proteins are created from the mRNA. So mRNA is it goes through translation. So it occurs in ribosomes, and tRNA brings anticodons, and eventually the codons code for various amino acids, which are what make up proteins. So amino acids join together to make the proteins. And mutations are changes in the nucleotide sequence, and some mutations have an impact and some don't. Those are called silent. And some are heritable, meaning they can pass from mom to child or dad to child. And these are usually mutations that occur in germ cells or in gametes. And then other mutations, I believe most mutations, I'm not sure if it's most, but mutations like cancer, they're not heritable. And frame shift mutations are when codons, that's 3 nucleotides, so the whole entire codon is changed. And there's a change in that base pair, that particular amino acids. That's frame shift. Yeah, so I will do the lab based on all this information. New share. Okay. Navigating Zoom, which is a little bit difficult. Let's see. All right. So hopefully that's good. So this is transcription. So we have to, you see DNAs from the 5' to 3', but remember RNA pull only reads DNA from the 3' to the 5', meaning nucleotides are only added from 3 to 5. So that's how we got to read it from. And the way I remember it is apple tree, garage car. So A goes with T, G goes with C, but this is RNA. So there's not going to be a T, there's not going to be thymine, it's only going to be uracil. So it's adenine, cytosine, guanine, thymine, and uracil, but there's no thymine, there's uracil. So let's start. T becomes U. T becomes A, sorry. A becomes T, but that's not T, it becomes U. C becomes G. And I'll pause the video and I'll finish it, and then you'll see.
Good afternoon, everyone. Today's topic that I will be talking about is the cytoskeleton and how it functions and why it is important for the cell. So, to start this off, the cytoskeleton is actually found in the cytoplasm of your cell, and the cytoskeleton helps the function of the cell in several ways, such as by giving it its characteristic shape. It also provides strength for the cell, and it allows cellular movement. The 3 types that we'll be talking about today are your intermediate filaments, your microtubules, and your microfilaments. So the first one that we will be talking about is your microfilaments, and out of the 3 that we will talk about today, these are the most narrow ones. And as you can see, it's kind of like a narrow structure of 2 proteins that are bound together, and these proteins are actually called actin. Now, actin can work with other proteins, and it can do things like providing muscle movements and even cellular division. It can actually bind with another protein called myosin. Now, myosin is actually really important because it helps with your muscle movement and your muscle contractions. And honestly, if some type of genetic mutation happens and the instruction isn't sent properly to make myosin, then that can form abnormalities, such as some type of skeletal muscle abnormality or even cardiac muscle abnormalities. And if you may or may not know, cardiac muscle abnormalities are actually really serious and can result in heart failure or even death. Microfilaments are kind of like rough, and they can disassemble, which allows the cell to kind of like change its shape and move around. And this allows cells like white blood cells to move to an area with an infection inside of you, and then it kind of like engulfs that bacteria through a process called phagocytosis. Up next, we have our intermediate filaments. Now, unlike your microfilaments and your microtubules, which we'll be talking about next, your intermediate microfilaments don't have anything to do with cellular movement, but they rather maintain the shape of the cell. It can also help provide mechanical support for the plasma membrane. Intermediate filaments are known for containing many types of protein, and one that you may know is keratin, which is found in places such as your hair, your skin, and your nails. Now, in this picture to the right, it shows you histology or tissue. The first thing that you probably see is something labeled desmosome. A desmosome kind of like connects 2 cells together, and you usually find desmosomes in your epithelia or your skin. Then it shows you the intermediate filaments kind of like going from the desmosome and then into the cytoplasm. And then for the last filament, we have your microtubules. Microtubules function in several ways, one of them being the transportation of membrane-bound organelles, and these can be found in your flagella and your cilia. Now, if you don't remember these terms, your flagella are your long hair-like structures and are kind of like used to move a whole cell by itself. An example would be sperm. And cilia are your short hair-like structures that can move substances on the outer surface of a cell. And in this picture to the right, this is actually cilia looked at through an electron microscope. Now, that outer thing that you're seeing is your plasma membrane, and then the inner ones that you're seeing are your microtubules, your cilia. So I hope you understand that the cytoskeleton is actually extremely important, and it takes part in many things in your cell, and how it's extremely essential for your cellular homeostasis and your survival. Thank you for listening.